In the second part of chapter 3, we're going to cover how molecules or substances can move into and out of our cells. Then we're going to go over mitosis and a little bit about cancer. Alright, so here, these can be for moving stuff both into and out of our cell. Passive transport. So here, the cell does not use energy to do this. There are three different types of passive transport: diffusion, facilitated diffusion, and osmosis. In each of these, we're going to see that the molecules are moving in a high to low direction. Okay, I'll show you what that means. Versus active transport. So here, these types require ATP, energy for the cell, to move the molecules into and out of our cells. And so different types, we'll see active transport. Endocytosis is when the cell brings in a large substance to the inside. So think endo, into. Exocytosis is when the cell spits out large substances or in large quantity. And then transcytosis, this refers to when the cell picks up a substance, carries it inside, and then exits out of another part. Okay, we'll start with diffusion. So this is passive transport. No energy is needed. Here, with diffusion and passive transport, substances go from high to low. This is also called down the concentration gradient. All right, so here we can underline from high to low. So again, no energy is required to move these substances. Simple diffusion, this is if the molecules or substances go in between the phospholipids. And facilitated diffusion is if a protein channel is used to move stuff either into or out of our cells. So now, just to make sure that you understand this, I'm just going to make a few drawings on the next slide. So again, this could be either for when the molecules or substances move into our cells or out. So let's say you just had a meal. So now pretend like these dots are food molecules. Well, you were hungry before you ate, and so your cells don't have many of these molecules. So when these molecules move into our cells, they're going from high to low. And so here, substances, molecules, have a natural tendency to go from high to low. Outside where there's more, they're being, think the pressure is too high. And so here they feel claustrophobic. They want to go from an area where there's a lot of them to where there isn't that many. But know that the opposite can also be true, meaning if you have a lot of some molecule on the inside, not so much on the outside, hey, again, these molecules have a tendency to go high to low. Okay, so diffusion is if these molecules are squeezing in between the phospholipids to go through the cell membrane, or facilitated diffusion is if a channel is being used. And so a channel would be used for polar molecules or a larger molecule. Osmosis is a type of passive transport. So here, this only involves the movement of water. But here also water, because it's passive transport, it also goes high to low. So we go from, here we can underline the word from, higher water to lower water levels. But yes, water goes high to low, but here, put double, triple stars by this. Know that water is also moving to an area where there is a lot more solutes. Here, solutes, this word means salt, sugar, and proteins. Water is attracted to these. So this is passive, no energy is required. All right, just to make sure you understand this, I'm going to make a few drawings of this osmosis as well. right? Let's say this is a cell. Let's say the outside of the cell has 80% water. Well, so in this liquid, there's water, but then there are also solutes that are mixed in. Let's say it's about generally these numbers. But the inside, now I'm making up these numbers, but let's say the inside has 60% water and 40% solutes. So this would happen as if you're drinking a bunch of water. Well, so now water is coming over to your cells. Here, this water that's on the outside is going to have a tendency to go into our cells. Why? Because it goes high to low. But notice, water is also going towards the higher solute region. And so here, proportionally, the inside has more solutes, and so the water is attracted to that. The more solute difference there is, the more solutes one area has, this osmotic pressure increases. Water wants to rush in with more force. When water enters our cells, it is said to be in a hypotonic solution. So with a hypotonic, here the cell gains water if there's more water on the outside. And when this happens, the cell swells. All right, we're going to see this word hypotonic on the next slide. But so what about the opposite? So the first one was if you're drinking water, all right, water's coming to your cells. But what if you're dehydrated? So now I'm going to switch up the numbers. I'll make this side 75% H2O, 25% solutes. Now we'll make the inside more water. Here we'll make it 80%. And of course, don't worry about these exact numbers. It's just to show the direction of the movement. Okay, so in this case, now there's more water on the outside. You don't have too much on the outside because you're not drinking enough water. Here, the water exits instead. So again, it's going high to low, but also it's going towards the higher solute region. Notice not as much of a difference here for the solutes. But here now, this one is called hypertonic. Here the cell loses water. When this happens, the cell shrinks. Well, we want the numbers that are on the outside and the inside, when you compare them, to be about similar. So let's say the outside is about 75%. And the inside is also about the same. When you have an equilibrium like this, yes, some water is going to trickle in, some water is going to trickle out, but it's going to be about equal. This is called isotonic, the way we want it to be. So here, no net gain or loss of water, and the cell remains the same size. By the way, with the hypotonic and the hypertonic, this movement of water is going to continue until the numbers on the outside is going to equal to the numbers on the inside, until equilibrium is reached. Okay, so the osmotic pressure, what I was talking about, is the degree or strength of osmosis. So here, know that with the isotonic, which is what we want it to be, this is the same osmotic pressure, meaning numbers are equal. So here you have no net gain or loss of water. Cell remains the same size. In a hypertonic solution, here the cells lose water. When they lose water, they shrink. In a hypotonic solution, here the cells gain water. And when they gain water, they swell. The way I keep these apart is to look in the words. With the hypotonic, it's got that O. So I think with the hypotonic, the cell is getting bigger and bigger, rounder and rounder. Think like when you fill up a water balloon. until it can actually burst. But so I think it's got the O, so the cell is becoming like an O shape. Versus with the hypertonic, so here this one shrinks. So think like it's hyper, it's just losing weight kind of thing. Hope that helps. Right, so diffusion, facilitated diffusion, osmosis, those were all passive transport. No energy is needed, and the substances go high to low. But what if the cell wants to move stuff low to high instead? Okay, so here we're going to underline from low to high. This is also called against the concentration gradient. Going high to low was down the concentration gradient, but now opposite, low to high, is against. Here with active transport, when you're trying to move stuff low to high, or we're going to see in just a little bit, when you move large substances or in large quantities, here you're going to use ATP, but also generally we're going to see carrier proteins or these special pumps, channels being used. So that's what we're seeing on the picture here. So here are these purple shapes. Those are the special channels, the pumps. And so these are moving the stuff low to high. Okay. Also know active transport needs ATP. So just to review for the active transport. So now this one goes low to high. And so this would be needed if you want to get these nutrient molecules to the inside. So you don't have too much on the outside. There is plenty on the inside, but hey, the cell still wants to bring these to the inside. You go low to high. This has to use some kind of a channel, special carrier protein like a pump. And here, this channel is using ATP or energy. Now, moving stuff using vesicles is also considered active transport. Endocytosis is if the cell brings in a substance into the cell using a vesicle. So here we can underline movement of substance into the cell. And so this is done if the substance that the cell wants to move in is too large to enter the cell. Or let's say you want to bring in a large quantity of these smaller substances. There are three types of endocytosis. Penocytosis, this is if the cell brings in droplets of liquid. Phagocytosis, this is for bringing in solid particles, which is what happens with white blood cells when they eat bacteria. And the receptor-mediated endocytosis, this is when the cell is being picky. It only brings in specific substances that are able to attach to these special receptors, which I'll show you on the next picture. So here in this third one, this shows the receptor-mediated endocytosis. On the surface of our cells are these special proteins, and here these purple shapes. So only the substances that are attached, that are able to attach these receptor proteins, are then able to be brought into the cell. Okay, so now the substances are contained in vesicles. Exocytosis is then the opposite. This is when the cell ejects substances to the outside. And so we'll just underline here, release of substances, particles from the cell, to the outside. Also here, now the vesicle moves to the surface of the cell. When it attaches to the surface of the cell, the vesicle opens and the stuff comes out. This is how neurotransmitters are released from neurons, or hormones are released by gland cells. Next, mitosis. And so mitosis is the division of the cell, but really it refers to the division of DNA. The picture here is showing the cell cycle. Cell cycle really means the life of the cell until it separates into two. And that's if the cell separates, is able to separate. The stages of the cell cycle, we have interphase. Interphase takes up the most of the cell cycle. Interphase is when the cell is doing its normal stuff. But also we're going to see on the next slide, This is when the cell makes 2 copies of DNA. Interphase has 3 parts to it. There is the G1 part. This is when the cell grows. The S part, this is when the cell makes 2 copies of DNA, does its normal stuff. Then G2, this is when the cell grows some more, does its normal stuff as well. But also, it's getting ready for mitosis. Then mitosis starts. And mitosis takes up, of course, less time than interphase. And so here we'll start with interphase. So again, like I said, here the cell grows, maintains its normal functions, but more importantly, here the cell replicates DNA, meaning it makes 2 copies of DNA. Okay, so this is very important. This is not done during mitosis, but during interphase. You want to do this during interphase so that if there are any mistakes of, when 2 copies of DNA were made, we can fix these in time for when mitosis starts. Okay, so the S part of mitosis, S part of interphase, this is when the DNA is replicated. And the G1, G2 is when the cell is growing. Okay, so then next for the different steps of mitosis. And so mitosis really refers to division of the nucleus, meaning the DNA, the 2 copies of DNA. Cytokinesis starts actually during anaphase. This refers to the division of the cytoplasm, meaning the splitting of the cell. Prophase, this first step of mitosis, this is when the chromatin condenses to form chromosomes. So here the chromatin is the long string form of DNA. So here the chromatin ends up finding its other copy that was just made. So then the chromatin ends up coiling to form X shapes. So here the 2 lines of the X's are the 2 copies. So here's 1 copy, here's the other copy. And so during mitosis, we're going to see the 2 lines of these X's separating into 2, since each line is 1 copy. Also during prophase, nuclear envelope and the nucleolus, those are going to break apart, and the mitotic spindle is going to form. During metaphase, the chromosomes are going to line midway. They're just going to line up in the middle part of the cell, also called down the equator. In anaphase, we're going to separate the 2 lines of the X's. So here the chromosomes separate, move in the opposite directions. The mitotic spindle, those spindle fibers, are going to pull these 2 lines of the X's, or the sister chromatids that are being pulled. Then in telophase, here we're going to see the cell kind of trying to split up into 2. Here the chromosomes return to the chromatin structure now that they have been separated. New nuclear envelope is going to form around them, so basically a new nucleus is forming in the 2 developing cells, and the nucleoli also become visible. Okay, so these pictures are showing the different stages. First one is showing the interphase. So here the DNA is in its long string form. Now I'm not sure if this is after or before the 2 copies are made. But so then in prophase, here the X shapes are forming. I know here the X shapes are not very easy to see, but here the chromatins found their copies. They attached together, and now the strings ended up coiling to make these chromosomes. Prometaphase is kind of the in between prophase and metaphase. But here now with metaphase, chromosomes are attached down the middle of the cell. So of course know that this can rotate. And they're attaching to these yellow lines, which are the spindle fibers. So these spindle fibers are really just microtubules, which are proteins. And so here they're attaching to the chromosomes. So metaphase for middle, they're lined up in the middle. In anaphase, the sister chromatids, meaning each line of the X, these lines of the X's are separating from each other because the spindle fibers, those microtubules, are pulling them away from each other. And now with telophase, here it looks like the cell is separating into 2. And a new nucleus is going to form around the newly separated DNA. Cytokinesis is a cytoplasmic division. So meaning the pinching of the cell until it's going to separate into two. It actually begins during anaphase. So here we have these special actin filaments, microfilaments, that help to kind of act like a belt to then split the cell. Okay, next a little bit about cancer cells. Now, normal cells, they're able to stop dividing when they become crowded. This is called contact inhibition. But cancer cells, they don't care. They want to keep on dividing, make more of themselves. So this is why tumors end up bulging outward. They become bigger because here, more and more cancer cells are being made. Also, one thing I wanted to mention is that some cells do not divide anymore. For example, neurons, they divide a certain number of times, but then stop. And so brain cells, after a certain age, we don't make more of them. Now, in some parts of the brain, yes, for example, the amygdala, but most neurons, they're not able to divide. Also, skeletal and cardiac muscle cells, they cannot make more of themselves until after a certain age. Smooth muscle cells, yes, but not skeletal and cardiac. There are two types of tumors. Benign tumor means that it stays in the same area, but a malignant one is able to travel to other organs using blood or lymph vessels. But really, it's the cancer cells that separate off from the tumor and are able to then travel to other parts of our body. Cancer is really the uncontrolled cell division, and it's thought to be, the cause of it is thought to be mutated genes. Now, two types of mutated genes that can lead to cancer. Oncogenes. Oncogenes are abnormal or mutated genes that increase mitosis. So when the cell should not be dividing anymore, these instead cause the cell to divide more. And so here the cell has become abnormal. Then the tumor suppressor genes, these normally limit mitosis, but once again, if they have become mutated or inactivated, removed, they cannot regulate mitosis. Okay, so cancer cells are called immortal because they just don't want to stop dividing. Normal cells, after certain divisions, after about 40, 60, they stop, but not cancer cells. All right, so then this picture is showing how the genes may be mutated, for example, the oncogenes, that can then lead to this uncontrolled cell division. Then this part of the picture is showing how the cancer cells can bud off from the tumor and travel inside blood vessels. And so this is also called metastasis. And so I'll write this larger for you. Metastasis means just the act of how cancer cells travel in blood vessels and go to other organs of our body and start cancer in those other parts. Apoptosis is the programmed cell death. Now it's actually a part of development, normal part of development. What happens is in the fetus, there is webbing between the fingers and toes. And so apoptosis is when these cells that make up this webbing end up dying. Now, apoptosis is also to get rid of abnormal cells. And so here the cell should go through this apoptosis if it knows that it's abnormal. And so here, abnormal cells normally go through apoptosis, but cancer cells escape it. So big part of cancer research is to see, hey, can we make these cancer cells go through apoptosis? But of course, it's not easy to turn this on. Necrosis instead means cell death from damage. And so this is not a normal process.